So today we have Mark Kopetsky from Organic Valley. So by way of brief introduction, Mark has spent his life in the dairy industry, but he's also spent it in the soil. He earned his BS and MS in soil science and went on to work as an ag agent for UW Extension for many years. In the past few years, he's been working as Organic Valley soils agronomist. So he helps the cooperative farmers get the most out of their soils, their crops, and their animals. He is an extremely knowledgeable soils agronomist, has helped me out in my graduate work and many other people that I work with. And as Harriet said yesterday, you feed soils to feed your plants. So today we begin at the beginning with the soil. Without further ado, Mark Kopetsky.
Well, thank you, Anders. I'm not sure all of that was deserved, but I appreciate it anyway. It's really nice to be here with you guys today, and welcome to all you who are joining us from a distance and watching the recordings. It's really neat what Anders has put together here, and it's just always amazing to me. You know, when I think way back in my early career with the university about the big chasm that there was between organic agriculture and the research community, and about how we made so many bridges with that. And so, Anders, you've had a lot to do with that, and thank you for doing that and for inviting me here to talk today. So, as Anders mentioned, we're going to talk today about soil fertility, specifically in organic cropping systems. And I know that a lot of you, particularly some of you who are online or listening to the recordings, are not necessarily organic farmers right now. So some of the things that I'm going to be talking about today may be coming at fertility from a little bit different perspective from what you're used to, and that's okay. There's a lot of different ways to look at fertility, and in the organic world, we take a little bit broader view than we often do in the conventional side of things. So we'll be going through some of that stuff, and I had a kind of a challenge trying to get everything that I wanted to cover today into a short period. So I'm going to try to get through the material that I've got. If at any point, though, and I'll try to leave some time for questions and discussion at the end. But if at any point, if I say something that just confuses you, by all means stop me, because I don't want you to go through from there on just wondering what in the world I'm talking about. Anders, could you do me a favor and maybe give me about a 10-minute warning before we have to close down? Thank you. Okay, and this is about my luck with technology here. I guess we can go up there.
You got it.
Yeah.
You can just hit the click.
Oh, there we go. Oh, great. All right. Okay, so I'd like to spend some time talking about soil testing, because when we talk about fertility, I believe that it's very important to know what the levels of fertility and your pH in your soils are before you start trying to manipulate something. And I'd like to talk about soil testing to begin with. Soil testing is a tool, and it's not a perfect tool. It's not good for everything in the world, and it's better at some things than it is at others. But it's still a very useful tool. It's not perfect, but it's still very good. And some things that we need to remember when it comes to soil tests is that every field on every farm has its own environment in it and its own set of fertility conditions. So it's important when you're doing soil testing to test pretty much all of your fields on a fairly regular basis, because what's going on in one field is not necessarily representative of what's going on in any of the other fields on your farm. So a soil test is only good for the area that you take those samples from. And it's only useful if the samples that you sent into the lab represent the average conditions in that area. And we'll talk a little bit more about that specifically here in just a minute. Another thing about soil tests is that fertility changes over time. It changes as we change our cropping patterns, as we put amendments on, as weather changes, as we get varying levels of precipitation, as temperature changes. So soil test results are not good for a long, long time, only a few years most of the time. So it's important not only to test most of your fields, but to do that fairly regular. And I usually suggest about every 3 years for most people. If you get to the point where you go through several cycles and the fertility is about where you want it, and it's not changing a lot, you know about what you have to do to maintain that fertility, you can probably slack off on that period a bit and maybe go to 5 years or even longer. But until you know how that field is behaving, I'd start with about 3 years as an interval. And I have on here, soil testing is best used in conjunction with forage testing, some sort of plant analysis, okay? If you guys are growing grain crops, maybe your emphasis isn't going to be on forage, but a little bit later on, I'm going to encourage you to include forages in your rotation. Forage tests are very easy, they're very common, it's not hard to do, they're not expensive, and so during that time in the rotation that you've got the crop growing, or I should say that you've got the field growing a forage crop, might be a good time to do an analysis on that forage. And I always suggest using a wet chemistry mineral analysis, not an NIR test. The NIR isn't very accurate when it comes to mineral levels. But if you don't want to test your forage, you can do a plant tissue test on the grain crops that you're growing as well. So the idea here is that we're using soil testing as our initial tool, as a way to take a look at what's going on in the fertility of the soil. And then we want to look at some kind of a chemical test on some plant that we're growing there to see how the plants are actually responding to that. And that might give us some information to modify the amendments that we're using that we thought of based on the initial soil test. Okay, so I want to talk a little bit about a good soil sampling technique. Now, the reason that this is so important is because we're testing such a minuscule amount of soil compared to how much soil is out there in that field that we're trying to get an understanding of. If you think about the weight of an average acre furrow slice, in other words, the weight of the soil across 1 acre, 43,560 square feet, down to the depth that we usually plow, approximately 7 inches, and excluding stones and things like that. And if we took that much soil and dried all the water out of it and weighed it, it's going to weigh somewhere in the neighborhood of 2 million pounds. That's on 1 acre. Most soil test labs, when they ask you to have a sample for them, would like you to send in about 2 cups of soil. So 2 cups out of 2 million pounds in 1 acre. That's what they're going to get. And then they're going to dry that down, and usually they grind it and sieve it. And out of that prepared sample, they're probably actually going to be testing a couple of teaspoons. So now we've got a couple of teaspoons of soil representing 2 million pounds in 1 acre. And most of the time, the areas that we're sampling are a lot bigger than 1 acre, depending on the field that you've got and the characteristics there. It might be several acres, and if you've got really uniform soil conditions and you've used a very uniform cropping system and your amendments have been uniform, maybe you're going to represent 20 acres or 40 acres. So it's a huge amount of weight that we're trying to estimate the characteristics of from a tiny little sample. So that means that the characteristics of that sample are very important. We want that sample to represent the average conditions in that field. So what that boils down to is taking a number of samples that we put together in a composite that we're actually going to send into the lab. When I say a number, what I'm talking about is usually about 12 to 15. 12 to 15 core samples that you would take down ordinarily to the depth that you plow. Put those in a bag or in a bucket, mix those all together very uniformly, and from that composite mixture, that's what you send into the lab. Any questions on that before I go any further? Okay, great. So with that, we go on, and I just want to talk a little bit about what are we interested in a soil test result. When we think about all the different elements that are there in the periodic table, there's about 17 of those that all plants need. And I'm going to pick on my alma mater here a little bit. I stole this chart from the UW Soil Science Extension website. And I'm very proud to be a graduate of the UW Soil Science program. But there's a problem with this chart here. And if you look at this chart, what it's showing here is all the ones that are shaded are supposed to be essential for plants, or at least beneficial to plants. When we talk about essential elements, what we mean is that every single plant on Earth needs some amount of that element in order for it to stay alive and to be able to grow. And if they're a plant essential element, they're either blue because you can get those from air and water. So hydrogen, carbon, and oxygen, plants need those, but they come from air and water. All the ones that are shaded in green are the ones that don't come from air and water that every single living plant on Earth needs to stay alive. And then there's some of those that are backgrounded in yellow. And what we call those is our beneficial nutrients. When we say beneficial nutrients, what we mean is that some plants may require those to grow, or we found that some plants will do better if they've got a little bit more of those, even if they can stay alive without them. So it's not quite as universal as an essential element. So we call it beneficial. So the problem with this is right in the middle there, you see cobalt backgrounded in yellow. Well, cobalt is actually an essential element. You know, we've known that for some time now. So it's just a little mistake. But at the time that this table was put together, it probably was thought of as a beneficial nutrient. And so the point of this is that as we gain understanding, as we learn more about plants, as we learn more about the world around us, our understanding changes and we learn new things. And so at some point down the road, maybe some of these other ones that are backgrounded in yellow, like selenium, maybe we'll discover that selenium is actually an essential nutrient and we'll have to change the color of that to green. So it's just something to keep in mind. So 17 of those that are essential for every single plant on Earth to grow, along with sodium and silicon and selenium that are beneficial.
You got a question from the slide previous about sampling. The question is, do you grid sample or walk the field and take some representative areas?
Okay, that's good question. There's a number of different methods, and what you want to do when you gather your samples, if it's a huge area, and you're going to be coming back, when I say huge, it's going to be a very good sized field. Sometimes people will grid sample there so that they can come back and they can adjust the rates of the amendments that they're putting on according to various areas in the field. So if you've got a variable rate spreader, or if you can just adjust, by spreading manure heavier or doing something like that as you're going out there, and you can monitor individual areas. Grid sampling is just fine. What we normally suggest for most people at the scale that most organic farming is done, we use the UW suggestion of walking across the field in a W pattern. If you're from Minnesota, you stand on your head and it's an M pattern. But in Wisconsin, it's a W pattern. Very important. So the idea is here that you go across pretty much the bulk of the field. And when you're gathering samples, make sure that you avoid anything that doesn't look representative of the average conditions. So if you see a wet spot, if you see a little knoll, if you see an area where there was a lane going through, or if you're out in a pasture, if you're close to a water point where the cows might gather and concentrate nutrients, if it's a place where you buried a rock pile, anything like that that's out of the ordinary. So it's a good question. Okay, so we talked about plant essential nutrients, 17 of those, but there were a lot of different elements in that periodic table, and does that mean that we could ignore the rest of those? Well, we talked about beneficial nutrients, so that's a question there. That's something that we probably ought to pay attention to. But there's also more living things in the soil besides just plants. We have bacteria, we have fungi, we have nematodes, we have arthropods and insects and earthworms and all these other critters that live in the soil. And they actually need more than those 17 nutrients or the 17 elements that plants need. So with organic farming, we want to look at things as a system. And we want to try to nurture the entire biological community that's in the soil besides just the plants. So it is okay for us to think about things besides those 17 essential nutrients. We want to feed all those things, and all those things that are in the soil have a purpose, and most of them are beneficial to our systems. I want to talk just real briefly about pH. I think that you guys probably know what I mean when I'm talking about pH. We're measuring either the acidity or the alkalinity of the system. A pH of 7 is neutral. If the number is less than 7, then it's acidic. If it's higher than 7, it's alkaline. And the further away we get from 7, the more pronounced either the acidity or the alkalinity becomes. And the reason that this is a big deal, if you look at this chart here, you can see these different plant nutrients. We've got nitrogen, phosphorus, potassium, sulfur, calcium, magnesium, iron, manganese, boron, copper, and zinc. Now that's not all of them, but it's the majority of them, okay? And the width of that bar, as you go across the pH scale, varies. And what this is meant to represent is where the width of that bar is the widest, where it's the greatest, that plant nutrient is most available. And what we see is that for most of the nutrients, as we get fairly close to a pH of 7, fairly close to neutral, that bar is fairly wide. Now we do have some of these micronutrients down here, like manganese and boron and copper and zinc, that are actually more available at the more acidic pHs. So what that suggests is that we want to be fairly close to neutral, but maybe just a little bit acidic to make sure that we've got all of the nutrients available that plants are going to need. And so every crop has a minimum pH that we need to observe in order for that crop to be able to thrive and to do well. And so if we look at the recommendations from Wisconsin about what the minimum pH is for these various crops, and I'm focusing here on grain crops, we can see that actually barley is the most demanding of the grain crops that we're likely to raise with respect to pH. Barley has to have a fairly high pH of 6.6 for that to be able to do well. Next on the list is soybean. We have to have at least 6.3. And as we go down from there, we can see that it gets less and less restrictive. Corn and wheat and triticale, we need 6.0. For rye, sorghum, millet, flax, and buckwheat, we can get by as low as a pH of 5.6. So it's important for us to think about this with respect to the grain crops that we're growing. And also, when you do your soil tests, if you do a soil test on a field the first time and you find out your pH is 5.8, that suggests to you that maybe barley shouldn't be the first thing you try to grow there, okay? Until you can get that pH corrected, work with something that will tolerate those conditions, and you can have time to improve that from there. But we want to talk, or we want to think more about just the grain crops that we might be most interested in marketing. Because with organic farming, we're not going to raise these grain crops in isolation. They're certainly not going to be done as monoculture. And hopefully you're going to be doing something a little bit more imaginative than corn and soybeans. Okay, you want to have a nice robust rotation. And so you have to think about all of the crops that you're likely to raise throughout that rotation. And it takes a while for us to adjust pH. Lime dissolves very slowly in the soil, and it moves through the soil very slowly. So this is a process that we can't decide that, in 2 weeks we want to have a different pH and make that happen. We want to be looking out probably a couple of years in advance if we want to change the pH. And because we'd like to have as many options in our rotation as possible, my approach is that let's try to pick a pH that we can work with that will give us as many options as we can for whatever we want to raise throughout that rotation. If we want to have alfalfa in the rotation, that's going to be the most demanding forage crop that we can have in there with respect to pH. We'd really like to have that pH of at least 6.8 and preferably not going above 7 for good alfalfa production. Red clover is a little bit more forgiving. If we can get it up to 6.3, red clover does okay. And bird's foot trefoil will survive and do okay at an even lower pH, if it's even up down to 6.0. Now, having said that, my own bias is that I would still like to get the pH up to about 6.5. because we find that at 6.5, we have a lot better biological activity in the system overall. And so if you decide differently, that's your business. You can do that. I'm just telling you what my bias is and why I'm looking at it that way. So I'm going to stop there to just check and see if we have any questions up to this point. Okay, all right, great. So when we think about the pH that we really want to aim for on a particular field, we've got a question.
We have a question from Leonard. And you can repeat the question after I ask it.
Oh, sure.
Okay. Is it beneficial or possible to front-end load nutrients into soils before transition to organic, this assuming some conventional nutrient applications are easier than organic ones?
Okay, so the question is it possible and or permissible to improve our soil nutrient levels while we're still conventional to prepare us for farming organically so that we don't maybe have to spend quite so much money? Yes, it is. It's both possible and permissible. And having said that, please don't think that you necessarily need to do that. You know, there is a mindset out there that, oh, you know, we can really improve our fertility using conventional materials with a lot less expense if we do it conventionally than if we do it organically. Well, maybe, but not necessarily. It's not always that straightforward. And in a lot of cases, we can really do the bulk of what we need to do following good organic practices that are just fine for organic certification too. It's an excellent question, and I appreciate that. So, okay, so I guess with pH, I just wanted to come back to this. Remember that you're not going to be growing your grain crops in isolation. So don't look at just the pH that you need to get by with your grain crops. Look at the overall rotation. And again, try to keep this in mind as an entire system. And the pH range that I usually shoot for is someplace between 6.5 and 7. Okay, so when we think about fixing problems in soils with fertility, we're talking about some sort of soil amendments. And there's a lot of different philosophies out there about how we amend soils. And I'm a simple guy, so I kind of take a simplistic approach to life. And the way I look at it is, you know, let me understand what the target levels for each of these nutrients are that I'm shooting for. And then I can decide how I'm going to approach that. Now, there's a lot of different philosophies out there in organic agriculture about soil fertility. And you'll have to investigate those and decide for yourselves which you think is the appropriate way to do it. We do have very good information that's based on research that's available from the universities in the various states about soil fertility. And that's a really good place to start, because the people who are living there and working with those cropping systems have had a chance to study those for a long time, and they've got a pretty good idea about what you're likely to find as shortcomings and the best way to correct those. So that's a great thing to rely on. There are also lots of independent consulting agronomists out there that you can go to get help. And there are consultants and dealers from various products that are selling products that will also give you some guidance. And just because somebody is selling something doesn't necessarily mean that they're going to give you bad information. So I want to make that clear. There are a lot of really good people out there who happen to be selling products. So you have to work with somebody who you're comfortable with, and I would suggest someone who has a proven track record, okay? Having said that there are lots of good people out there, there are also occasionally people who are misled and who I think are probably giving advice that's not helpful. So, you know, take a look at the advice that people are giving, take a look at the results that the farmers who they've been working with have had, and come to your own conclusion and find somebody that you're comfortable working with. It's perfectly fine also to rely on farmer experience. So what's going on in a particular region where farmers have been working with the system for a while and they found things that work, that's really valuable, you know, and don't discount the value of that. There's nothing wrong with farmer experience. So if you know what your target is, you have an idea how to shoot for it. So what I like to do then is once I know what my target levels are, when I get my soil test back, I can compare each of the parameters on the soil test report to what I think those numbers ought to be. And then the areas on the soil test report where there's the biggest gap between what I would like to see and what's actually going on the ground seems like a really good thing to work on, right? You know, if I work on those things, if I work on the big glaring gaps, the big holes, I'm going to be able to see a lot of progress fairly quickly. And another thing about that is that when you have a huge difference between what you want and what is actually there, everything that you do to go toward that goal is going to make a huge difference, and it's really gratifying. You know, it pays its way very well, and it's also good for morale because you can see that you're making a difference. The closer you get to what you want, the less of a difference you're going to see as you keep adding these things. Some of the common things that I see on the soil reports that I review for our cooperative as being places where people can work on are things like pH. You know, in quite a few cases, we have soils that are too acidic, but we also do find soils that are too alkaline. People who have been putting poultry manure on from laying hens for a long, long time, it's got a lot of lime in it, that can bring the pH up. Some places in the western United States, the pH is just high to begin with. Yeah. Question?
Really the question. The farmer says that he has a high pH soil, 7.7. And so if there are very high pH soils, what kind of effect is that going to have on common crops?
Okay, well, that's an excellent question. And we'll get to that a little bit here in just a minute. But in general, what happens is that high pHs tend to be associated also with very high calcium levels quite often. And it's interesting that when there's a sort of a magic range of most nutrients for plants. And when we're within that range, the system works pretty well. And if we're short of that nutrient, things don't work as well. But also, if we're long on that nutrient, and in this case, for instance, calcium is quite often very high in soils that have a very high pH. Interestingly, when that happens, for some reason, the plants stop taking up as much calcium as we'd like them to. Even though they're basically drowning in calcium, it's like, I guess it would be like, you know, if you like chocolate cake, and, you know, you go into the bakery, and you've got free chocolate cake, and you start eating chocolate cake. Well, geez, after a while, you know, you get tired of chocolate cake, and you're just not going to eat anymore. I don't think plants look at it exactly the same way, but that's just kind of the way I look at it. Too much of a good thing is not good either. Another thing about high pHs, as we'll see here in just a few minutes, is that it tends to bind up a lot of the micronutrients. And micronutrients we'll talk about more too, but micronutrients are very, very important. A lot of times, it's easier to amend an acidic soil and bring the pH up than it is to try to bring the pH down on a high pH soil. So that was a great question. Thank you. So pH is one problem that we see very commonly. The other ones too, you know, you've probably heard people say organic farming is about more than NP and K, and it is. But NP and K are still really important. You know, we've got to have a lot of nitrogen. We've got to have a lot of phosphorus. We've got to have a lot of potassium. And so if we don't have those things that we need in the system, the crops are going to suffer. So we have to pay attention to that as well. Next on the list, we have things like calcium and magnesium, and there's a lot of different philosophies out there about calcium and magnesium, and people get different guidance from different people. I would just say that in the majority of the cases, when we look at calcium and magnesium, from my perspective, it's not necessarily a case where we don't have enough calcium to provide calcium as a plant nutrient. But calcium is also important in forming soil and maintaining soil structure. And that plays into soil quality. We want to have well aggregated soils. And so there is a very good reason that we want to have enough calcium. But as we just talked about, too much of a good thing is not what we want either. Magnesium often gets a bad rap because a lot of people feel that most of our soils have way too much magnesium. In some cases, I think that's true. I think it probably gets a little bit more attention than it deserves for the average conditions that we see. But magnesium is also a very important plant nutrient. The central atom in every chlorophyll molecule is magnesium, kind of like iron in our hemoglobin in our blood. It's very, very important that we have enough. So we have to take a balanced look at this and consider all the things that go into fertility. Potassium is also very important, and I tend to look at the potassium numbers on a soil test result that are extracted from the sample, not the percent of the base saturation. I'm not sure if we're going to have time to talk about base saturation a lot, but basically, when we talk about percent base saturation, what we're talking about is the proportion of calcium and magnesium and potassium and sodium that a soil is holding on its exchange capacity. Okay, so with calcium and magnesium, I think that's very important to look at. What proportion of those? With potassium, I would rather look at just what's extracted from the sample because that's what the plant is sensing. The plant doesn't care if it's 3% potassium or if it's 7% potassium. The plant cares if it has enough potassium to be able to grow well. Oh, and then we've got sulfur and boron. Sulfur and boron are things that are not held very well in soils from year to year, and it has to do with the electrical charge of the ions. Things that have a positively charged ion, which we call cations, which are calcium and magnesium and sodium and potassium, also hydrogen. Hydrogen causes acidity. But those things that have positive electrical charges are held on soils very well. Things that have a negative charge, like the boron, the borate molecule, the borate ion, and the sulfate ion, are not held on soils very well from year to year. But plants need some of these, and we're always extracting those when we take the crop off of a field. So because the soil doesn't hold on to them very well, and because we're harvesting those every year, it's usually best to add some of those every year. And that's true whether you're growing grain crops, forage crops, forage crops for hay, pastures, whatever it is. Those nutrients are still very important, and it doesn't take a lot, so it's not going to break the bank. Okay, and then we've got other micronutrients involved too, things like zinc and manganese and copper. There's also things like molybdenum and some of these other stray cats out there that we just never test for. We never test for chlorine in Wisconsin because we've never seen a shortage of chlorine. When you get out west, that's different. The crops out there in those conditions can show chloride deficiencies. But in our area, in this part of the country, we just don't see that. We don't test for things like molybdenum because it's present in such tiny amounts that, it's really hard to find. And if we do find it, we don't have interpretations to go with those numbers. So just a few things to keep in mind there. Okay, all right. There's a lot of different labs out there, and you'll see lots and lots of different formats for reporting soil tests. This is one that we use with our co-op, and we put it together to try to simplify things a little bit. And there's all these colors on here. What this boils down to is that if the number that we see for that parameter in the soil is lower than the target range that we would like, it shows up as yellow. If it's within the range, it's green, and if it's above the range, it's red. So fairly straightforward. Just one example. There are other companies, soil consultants and product companies that do things along these lines too. Sometimes you just get a piece of paper with a bunch of numbers. So whatever lab you're using, familiarize yourself with the results and understand how to find what you're looking for on those. Okay, thanks, Anders. Okay, I would like to talk a little bit about nitrogen, and I should back up because I want to show you something here. This soil test report, it happens to come from a private soil testing lab out in Omaha, Nebraska called Midwest Labs. And this lab runs a nitrate analysis on every sample that they get in. The UW lab does not do that, and the labs that are following the UW recommendations don't do that. There are a lot of labs that do this routinely. And the point that I want to make with this is that if you do get a number for nitrogen on your soil test report, and it's a routine sample that you sent in, you can safely ignore that because it really isn't very useful. Nitrogen changes forms in the soil all the time, depending on the time of the year, depending on the crop that was growing there, the crop that is growing there, the temperature, the moisture, all these things. Nitrate is always changing forms back and forth. And the labs that test for nitrate typically are typically only testing for nitrate. So you are getting an accurate representation of that single form of nitrogen, but it's not very useful. If you want to test for nitrate, there is a very well-defined procedure that you follow, and you do it at the right time of the year, and you do it to the right depth, and you do it under very controlled conditions, and you get the sample to the lab very quickly. And there are good ways to employ that. We call it a pre-plant nitrate test. But this isn't the test. And so when we get the nitrate numbers here from Midwest Labs, we just ignore those. We don't even show them on the report. But nitrogen is very important. And it's especially important to grain crops because most of the grain crops that we grow don't fix their own nitrogen. Soybean is an exception. Lupins, if anybody gets into growing lupins. But a lot of times when people are farming conventionally, they focus a lot on nitrogen. It's easy to get nitrogen, and it's easy to dial in the amount that you want if you're farming conventionally. When we're farming organically, we have a lot fewer options for artificially bringing nitrogen into the soil. But we do want to make sure that we've got enough nitrogen there for the plants to grow. And so I've got this slide here that just shows you relative amounts of nitrogen that we would need to have for various crops. And these come right from the UW recommendations. So if I was growing conventional corn, depending on the price of corn that year and depending on the price of nitrogen fertilizer and depending on my soils, and if it was a sandy soil and I was irrigating, they might ask me to put on up to 250 pounds of nitrogen per acre that year to grow a good corn crop. Most of the time it would be less than that. That's an extreme. Most of the time in Wisconsin, we're probably looking at between 120 and 180 pounds of nitrogen per acre for a conventional corn crop. It's got to come from somewhere, okay? Sorghum is a first cousin of corn. We really don't grow corn, we don't really grow sorghum for grain here because we just don't get warm enough most of the time. But if we were growing sorghum, I know we got some southern guys with us too, so I wanted to include you guys here. If we are growing sorghum, sorghum is a first cousin of corn, but it doesn't need quite as much. So we can get by with less nitrogen for sorghum. Wheat, amongst the small grains, wheat is right up there. Wheat takes a fair amount of nitrogen, up to 105 pounds per acre. Sunflower can also be a real heavy feeder. You know, there are a lot of organic sunflowers out there. And that takes a fair amount of nitrogen, you know, probably about half of what you'd want for a corn crop. Canola and millet and rye can also use quite a bit, you know. And then as we go down the list there, we've got oats and triticale, and then down to buckwheat and flax, and then soybean at the bottom there. And our official suggestion is 0, but we know that there are people who put nitrogen fertilizer on soybeans because it's just such a wimpy crop. Sorry, I had to expose my bias there. But anyway, so you can see that there is this hierarchy. That nitrogen has got to come from someplace. It doesn't necessarily have to come from fertilizer. And in organic farming, what we would like to do is we would like to work with the biology of the whole system as much as possible. And that biology can provide us with the bulk of the nitrogen that we need to get a very good corn crop or any kind of grain that we're interested in growing. So we want to start by focusing on biological nitrogen fixation from legumes. So what this means is that in your rotation, you want to make sure that you do something to grow a leguminous crop in there. If you're going to strictly grow grain crops, you know, you're looking at things basically like soybeans and peas, probably. Those are the two most commonly grown. They're not technically grains, because grains are actually seeds of grasses, but we think of them as grains. But those are things that could be in a grain rotation, that are legumes that will provide some nitrogen. Now, those are not going to provide anywhere near as much nitrogen as a good plowed down hay field with a nice bunch of alfalfa in it. So that's kind of like the gold standard. So if there's a way for you to incorporate forage crops in your grain rotation, you know, we have a demand for organic hay too. So think about that as you're setting up your rotations. And if your land is such that you can grow good alfalfa because you've got the drainage and the pH is up and the fertility is up there, that's a really nice thing to have in a rotation. We can also look at things like livestock and poultry manure. There's a lot of nitrogen in that. And then there's things like blood meal and feather meal and fish fertilizers. There is a product, sodium nitrate, that's actually mined that we can use in organic farming. We tend to try to avoid that as much as possible. It's a very, very soluble product. It's got a fairly high salt index, and we can do better than that. You know, it might have its place just as a supplement, just as a starter form of nitrogen or something like that, or to give you a little bit of an extra bump, something like that, but we don't want to rely on that. And then there's foliar feeding and things like that that are also good sources for micronutrients if you decide to do that. Even if we don't have a good hay field or something like that to plow down, even if we're just rotating a crop from one thing to another, we do get a little bit of a nitrogen credit from that. It's not really that crop is necessarily making nitrogen, but plants have a certain amount of nitrogen in them as they're growing because plants have some protein, and protein is a component of, nitrogen is a component of protein. So when we plow down any kind of a crop, we're going to have some kind of a nitrogen benefit from that. So that's what we call rotation effect. It's not that they're necessarily accumulating more nitrogen. Okay. So this is a chart from a very nice publication that the UW Nutrient and Pest Management program puts out. The publication is just 2 pages front and back. It's called Nutrient Management Fast Facts, and it's free. I've got a few copies for the people who are here today. If you haven't seen this already, I'd be happy to give those to you. You can also order these for free. Just Google it and you'll come up with it, and there's a phone number on there. But there's some really nice, handy information here. We don't have time to go through this in detail, but you can look at this at your leisure. And the point with this is you can get a substantial amount of nitrogen from plowing down a hay field. So if you've got a good alfalfa stand and you've got some regrowth there and it's on a loamy soil, you can get up to 190 pounds of nitrogen from plowing down that alfalfa field. And that's more than you need for most corn crops. So it's good. It's worth looking into. Manure is another source. Okay. And again, we don't have time to tear this apart in detail. But any kind of livestock manure or poultry manure is going to have a fair amount of nitrogen in it. It's also going to have phosphorus and potassium. And that's important because depending on the soil test levels of the field that you're putting this on or that you want to grow the crop in, that needs nitrogen, you may or may not be able to benefit from the other nutrients that manure has. So this is best as a nitrogen source, as the main nitrogen source on fields that also need extra phosphorus and also need extra potassium. So again, we want to include legume crops of some sort in that rotation. And the sequence that you grow your crops is very important. So if you've got a crop that's going to provide some nitrogen and you have other crops that are going to need nitrogen, grow your highest nitrogen demand crop after the crop that makes the most nitrogen so that you can take the most advantage of that. In other words, if you have an alfalfa field and you're also going to raise corn and soybeans and, let's say, wheat Okay, we already talked about soybeans don't really need any extra nitrogen. Wheat's kind of intermediate, and corn is the highest. So that suggests that your rotation ought to go in the order of alfalfa, corn, wheat, and soybeans. Does that make sense? Okay. And then if we need more nitrogen than what we can get from the legume credits, that's when we come in and we supplement the rest of it from manure or from some other source. So take advantage of the stuff that you can do on your own with the biological system first.
So the surface could have a few more minutes, and we'll hear if you guys have any questions for you.
Okay, when do you guys have to take off?
In about 4 minutes.
Okay. All right, we're going to fly through here. I just wanted to show you on here, this column, let's see, that column is phosphorus, and this column, for you guys who are remote or watching the recording, that column is potassium. And so one of the reasons that we came up with this format is when we want to spread manure on a field, the best places to go with manure are where both the phosphorus and the potassium columns are yellow. It's just an easy way to do it. It's just an easy way to visualize that. Okay, I do want to talk to a little about micronutrients and minor nutrients. I mentioned about boron and sulfur already. Those are both very important to every crop, and especially important to legumes because they're both involved in biological nitrogen fixation. So if we don't have the boron and the sulfur out there that we need, we're also not going to make as much nitrogen as what we'd like. So again, think of this in the big picture. And micronutrients are those things that we need some, things like copper and zinc and manganese, even iron, cobalt, molybdenum, all those other ones, we need to have some of those available for plants to grow, but they're tiny amounts, okay? And when we think about the micronutrients, there are some of the grain crops that will require proportionally more of some micronutrients than others. So on the next slide here, we've got corn. Corn is famous for needing a little bit more zinc than a lot of other crops. Soybean and wheat and oat, manganese is very important. With canola, it's boron, both boron and sulfur. Canola behaves kind of a lot like a legume because it needs a lot of both of those. Question?
Can you just go to your co-op and get zinc, manganese, boron bags? Where do you get that stuff?
Well, very good question. And for most of those, like say for sulfur, the common sources of sulfur that we would look at would be gypsum, calcium sulfate. If the pH is high and the calcium is high, we probably want to use some elemental sulfur. So just plain sulfur in that form, that's not soluble and it's not available to plants. But once we put it in the soil, there are various types of bacteria that will use that sulfur as a food source, and they convert it to sulfuric acid and make it available to plants. With a lot of them, zinc, copper, manganese, the sulfate form of those is the form that we can use with organic cropping systems, and most soil amendment dealers do carry those. With boron, it's usually either borax or calcium borate or commercial products like Solubor or Granubor. Those are all acceptable for organic cropping systems. Did that answer the question? Okay, great. Before anybody leaves the room, you have to successfully reproduce this diagram and give it to Anders, who will check them all over on your way out. Don't worry about the details on this. You can take a look at this at your leisure. But what this shows, this is called Mulder's chart. So far, I have not been able to ascertain who Mulder was or is, but I got this off the internet, so it must be true. This is something that you'll see at a lot of organic soil fertility meetings. And it's used not just with soil fertility, but also with animal nutrition. And what it shows is how a lot of different chemical elements are interrelated to other elements. And so the point behind this is that we never deal, just like we don't deal with a grain crop in isolation, neither do we deal with a plant nutrient in isolation. When we try to adjust one thing, or if we're short or long on one element, it's also affected and affecting other things that it has an association with. So that's all that I wanted to make the point with that. This is just, I'm not going to go through this because I know you guys have to go, but this is something you can refer back to later on. It's just a list of some common fertilizer products that are fine for organic cropping systems, and you can look at that as you have an opportunity. Again, we're focusing here on soil fertility, but don't neglect the other characteristics of the soil too. You know, the physical characteristics of the soil are very important. Biology in the soil, and all these things are interrelated. So it's important to take a look at all of that stuff. Again, when we think about fertility and you identify some deficiencies, you know, come up with a sort of a hierarchy that you can follow for what amendments you're going to use that are going to do you the most good the quickest. So focus on those fields that have the biggest problems, or those components of fields that have the biggest problems, and try to work on those first. Everything is important, but you'll get the best benefit the quickest by trying to fill in those biggest holes first. And then your crops are going to do better, you're going to get more money in your pocket, and you're going to have more opportunity to do the fine-tuning down the road. And it's perfect. Okay, all right, very good. Well, I appreciate you guys coming.
It's Monday, quiz day. Not all of that W, there was an internal error. Not Monday, Tuesday. Okay. Not Tuesday. I'll be here on Monday. And there's still 20 people online, though.
Okay, all right. We'll keep talking until the last one drops off here. Okay. I did want to mention, too, you know, again, foliar feeding is an option. And foliar feeding seems to work the best for situations where people have most of the basic soil fertility already taken care of. But it is an extremely effective way to deliver micronutrients, too, especially things like manganese. Manganese does not behave very well in the soil. So we can identify shortfalls with fertility in manganese, and we can apply manganese. We can put it on in a form that's soluble. Manganese sulfate is soluble. It should work fine, and we put it on, it dissolves, and it gets bound up with something else. But if we put that directly on the plant, it's very effective, and it will get to the plant, and it will produce the results that we're after. So keep that in mind. And another one, too, is just strive for diversity. You know, we want to have as complex of a rotation as we can practically have, and we'd like to have as many different types of plants growing on one piece of ground over a period of time as we can, because every plant has its own characteristics, its own needs that it has for fertility, and it also contributes different things to the soils. So we'd like as much of that going on as possible. Okay, and I just wanted to mention too, you know, don't get too bent out of shape if you do some soil testing and the report comes back and it looks horrible. That's just an opportunity to make things better, because you probably didn't create those conditions. You know, you're probably just finding out about those, so you don't necessarily have to feel responsible for that, but it gives you an opportunity. And when you start working with a soil like that, it's very responsive. So you'll see great improvements with just a little bit of effort and not a whole lot of investment. Another thing about this is that even when you see big deficiencies, try to be patient with this and just correct those in modest doses over a period of time. You know, it's much better to put on a modest amount of a fertilizer or lime and work at that over a period of time than it is to try to correct that all at once. And you don't have to become a soils agronomist to be able to work on your fertility. If this isn't something that you have a strong interest in and you just don't want to mess with it, there are people out there who can help you. And those are some references that I included there. The UW has some really nice soil fertility references. There's also this book from the SARE program. It's one of my favorites here, Building Soils for Better Crops. And Gary Zimmer, who's associated with Midwestern BioAg, has a couple of good books out. This one, The Biological Farmer, He's got some really nice discussions in there about various things having to do with fertility. So I believe that is the end of mine. So if there are questions, I can hang around as long as people are interested.
There's a lot of questions online. I'm curious, you kind of went over the micronutrients, this slide, or soil emissions.
Okay.
I'm curious if you want to talk about that a little bit more.
Okay, sure. When we deal with organic cropping systems, we have some limitations for what kind of fertilizer materials we can use. Most types of lime are just fine. The exception to that is if people are using a product called kill dust, which is basically an industrial byproduct where an industry is taking limestone and they're heating it up to very high temperatures and purifying the calcium carbonate or doing something with it. But what they end up doing is the spent product of that comes out having a lot of calcium oxide. Calcium oxide in the presence of carbon dioxide in the water eventually turns back into limestone over a period of time. So it'll go from calcium oxide to calcium hydroxide and then finally to calcium carbonate. That takes some time. We'd prefer not to get calcium oxide on the land because that's prohibited by the National Organic Program. So we can't use kill dust as a lime source. On the bottom of the list there, wood ash is one that we can use. Interestingly enough, wood ash also has some calcium oxide in it, but it doesn't hang around as calcium oxide very long. Most of the time when we get wood ash, we've already applied some water, and so that chemical transformation is already underway. And wood ash is a pretty natural product. The philosophy, the starting philosophy behind what is okay for organics and what is not okay for organics starts here. If it's natural, it's probably okay. And if it's synthetic, it's probably going to be prohibited. And there are exceptions on both sides of that. But that's kind of the starting part of the thought pattern that goes into this. When it comes to phosphorus, no conventional farmer that I am aware of is still using rock phosphate because it's very insoluble. It has a lot of other stuff riding along with it that most people would consider to be impurities, things like calcium and sulfur. Well, calcium and sulfur are really not bad. That's just fine if we put that on there. But when we look at phosphorus fertilizers, if we're going to use an artificial amendment, it's probably going to be rock phosphate. Okay, and it takes a while for that to become available. It's easier to dissolve when the pH is very low, and we've got very acidic systems, but we don't really want that. But having a lot of legumes in the system, for instance, having an alfalfa hayfield or a red clover hayfield, those legumes will draw off a disproportionate amount of calcium. And when they take the calcium out of the system, that helps the rock phosphate to dissolve also. So it's not a useless soil amendment. But if we can tolerate the amount of nitrogen and the amount of potassium that we'll also get when we put manure on, manure is a very effective way to get phosphorus into the soil and have a fairly quick response from it. So those are the things that we look at with manure. With phosphorus. With potassium, our main products again are potassium sulfate and sulfomeg or K-meg, which is potassium magnesium sulfate, and again, manure. Those all have potassium in it. And I don't want to read the list, but if anybody has specific questions about these, I'd be happy to address those. So I don't know if the people online were...
So I'll stand up here with you,
okay.
And we can read through the questions.
Okay. We got it down there. Okay, all right.
Yeah. So we had the front end, high pH. Do you use or know of a linear programming type model that has been created to assist with crop rotation decisions? It would take into account your nutrient availability, plant needs, and amendment prices. It would employ the same logic as a dairy cow ration.
I am aware of a company that was planning on doing something like that. I don't use that myself. because when I get a soil test result back, we have about 1,800 farmers in 36 states right now, and I honestly can't keep track of the prices of amendments in all those places. That could be a very good tool, and if somebody can monitor all those things and they can put that in, I think that might be very useful. I know there is a company here in Wisconsin that is looking at doing something like that, but you have to have an awful lot of information, and for the volume of reports that I get back from a wide geographic area, I'm not able to do that. So let's see. Yeah, so I hope that answered the question.
Okay, and they can type a follow-up if they have more questions. So how would you, for example, get rid of excess calcium? That's what we're talking about, basically.
Okay. When we talk about different nutrients in the soil, when we have things that are cations, in other words, things that have a positive charge, we're talking about calcium and magnesium, potassium, sodium, and hydrogen. Also things like iron and zinc and manganese and copper. Those are a tiny little bit of the proportion of the total soil. And what we find is that these things that have these positive charges are also held to different degrees by the soil's exchange capacity. And what we find is that calcium is held the tightest, followed by magnesium, followed by potassium, followed by sodium, followed by hydrogen. So it's real easy to get hydrogen off of the exchange capacity of the soil, and it's really hard to pull calcium off, but we can. But we don't look at trying to correct excessive amounts of calcium by trying to flush it out of the soil, because that just isn't practical. We'd have to get rid of so many other things first to get to the calcium that it would just really disrupt the system. So my philosophy on that is, rather than trying to get rid of the excess calcium, let's work with the system and supply the other nutrients that we need for the crops to grow well, and start harvesting the crops from the ground, so that over time we're drawing those levels down that way. But it's just tough to get the extra out of there when it comes to calcium. So I hope that answered that question from Nicole.
Wendy, your opinion on wet versus dry soil test sampling?
Okay, that's a really good question. When you take your soil samples for testing, it's best if you can pick a time of the year, say for instance spring or fall, and follow that same time of the year every time you sample from then on. I'm partial to fall sampling because in the fall of the year, we've gone through the cropping system. The crop that we're growing there has extracted the nutrients that it's going to take out of the soil, and we're basically getting ready for the soil to almost go to sleep. That's not exactly true, but we're not going to be using much for nutrients through the winter. The other nice thing about sampling in the fall is that if we need to buy a lot of inputs, it gives us some time to do some shopping. And quite often we can get discounts if we buy things in the winter. So those are reasons. With respect to wet and dry, I like to focus on just an ordinary moist soil condition. If things are too wet, it's possible that we can leach some things out. Most of the things that we're testing for in soils don't really leach that much, but we can move potassium down, and that would affect that. On the other extreme, when we sample dry soils, sometimes we find that we've got more potassium showing up in the sample than what we would really think is there, or what's really going on in the real field conditions. And apparently what's going on there is that as the soil dries out, plants are wicking up water from down below in the soil. And it's also evaporating, just going out the surface of the soil. So that water is carrying behind it things that are dissolved in it. Potassium is one that's fairly soluble. That can come up to the surface, and as the moisture leaves, the potassium is sort of concentrated there. And of course, after it rains again, it's going to get dissolved and go back down in. But if we take a sample then, where that potassium has been brought up to the surface, we can see some artificially high levels of potassium. So that's a great question.
Next one from Mark Dudla, and that is, for weed control, we are now looking at water-soluble calcium levels less than 100 parts per million for weeds. The goal is 300 parts per million, far fewer weeds. What are the best cover crops to solubilize the lime that has been applied?
Okay. Well, I'm going to expose my ignorance first of all and say that I'm not real well acquainted with the water-soluble testing philosophy. I know that there are some consultants who use that a lot. And if farmers are working with that system and they're having good luck, that's terrific. I just don't have a lot of expertise in that. But if you want to get calcium out of a system, probably focus on legumes, and especially hay, because you're going to remove huge amounts of plant nutrients when you take hay off of the land. So when it comes to cover crops, look at a mix of cover crops that has a good, strong legume component in it, and actually, instead of just plowing that down into the ground, which is going to simply recycle the nutrients that are there, you have to remove that and get it out of the system. And that's probably about the only practical way to bring calcium levels down. You're not going to leach calcium. It just doesn't work that well. But you'll have to physically accumulate it. I should say you'll have to accumulate it in the plants and then physically remove that from the system to get it out of there.
Can you talk about, so do you have thoughts about calcium levels and weeds? So you're talking about using calcium.
I'm not acquainted with the research that would be behind that, and it could be true. I'm not saying that it's not, but I'm not aware of that.
Okay, that makes sense. This one's from Bruce. Because my soils have high levels of P, I'm unable to apply turkey poultry litter. Is there any way to tie up that P by, say, composting so I can use the litter?
No.
Short answer.
No, just say no.
All right, and then here's another question, which is, in your alfalfa, corn, wheat, bean rotation, where does the N for the wheat come from?
Okay, that's a good question. That's coming from the alfalfa. And the background behind this is that alfalfa is a typical legume. In other words, it has an association with a species of bacteria that infects the roots. And it's a beneficial relationship for both the bacteria and the alfalfa. The alfalfa gives the bacteria a home, and it also provides it energy. The bacteria has an enzyme in it called nitrogenase, which it uses to take nitrogen gas out of the atmosphere. We're almost swimming in nitrogen in our atmosphere. 78% of the air that we breathe is nitrogen. That's an inert gas. So we breathe it in, we breathe it out. It doesn't help us, it doesn't hurt us. Plants need a lot of nitrogen. And these bacteria are able to take the nitrogen from the air and convert it to a form that they actually use to build the amino acids that they build their bodies from. And they make more than what they need, and they also furnish the alfalfa with nitrogen fertilizer. That comes at great cost to the alfalfa plant because it's a very energy-intensive process. But it's a good deal for the plant because now it's getting nitrogen fertilizer. So we have a lot of nitrogen in a system that's got a lot of legumes. When we plow that crop down, that nitrogen can decompose and it can become available as a fertilizer to the crop that follows that. So that's where the nitrogen from the corn is, or excuse me, for the wheat is coming from in that system.
Wendy, do you think our technology can accurately test micro levels?
Yes. The question is, I don't guess I have to repeat it because you're right here. Can our technology accurately test micro levels? When it comes to things like zinc and manganese and boron and copper, we're pretty good. When it comes to things like molybdenum and selenium and cobalt, most labs don't do that. You can find labs that will test for that, but the problem is we don't have any correlation numbers. So we get a number back, say we test for cobalt. We get a number back, and we really don't know how to apply that number. So we can do that, but I don't see the value in doing that. With zinc and and copper, the results that we get from soil testing are fairly applicable to what we would expect to see going on in the plant. Manganese, even though it's an accurate test, manganese is such a squirrely nutrient that we can see levels of manganese in soil tests that make it look like, we're really not going to get enough of that, but the plants do just fine. And so here again, it's an example about why we would want to use both soil testing and some type of plant tissue testing to get a better picture of that. I thought there was one more thing I was going to say about that, but it's escaping me right now.
If it comes back, we can revisit.
Okay.
And this is a response to Bruce,
okay,
from someone at Purdue, giving a link to a talk, which I think was by Mirsky, a researcher in the Southeast. So that's there in the chat box if you guys want to follow that link. It's a YouTube link.
Okay, great.
Do you have time to talk about P and K organic options?
Sure. Here again, any kind of livestock manure or poultry manure is going to have a fair amount of phosphorus and potassium. And it's usually, I shouldn't say usually, but often it's fairly inexpensive. So if you can use manure in your rotation, that's just fine. The other things for phosphorus, basically what we're looking at for phosphorus is rock phosphate. That's about our options. It's either manure or rock phosphate. With potassium, we've got manure. We've also got potassium sulfate, sulfomag or K-mag, which is potassium magnesium sulfate. There is a mineral out there, or I should say there's a rock out there called green sand that has a mineral called glauconite in it that some people use. I don't suggest doing that because you're going to go broke. You're going to be putting on huge weights of that to try to attain a level of potassium. So that's really not practical. Wood ash, I mentioned wood ash being a good liming agent. Wood ash also has a substantial amount of potassium in it, and so that's another source for that. But that's pretty much it for P and K.
The follow-up that I have to that I was curious about is what trials have been done. I should probably know this as a researcher, but what trials have been done in terms of the effectiveness of nutrient availability between pelletized chicken manure, composted product, different kind of poultry manure, liquid dairy versus deep-bedded packs. So manure is not manure to a certain extent.
That's right. That's a really good point too, Anders. Manure is not manure, and I think even more important than just differentiating between sources, if you're going to buy a commercial composted product, like say composted poultry manure, quite often they'll have the analysis done on that, so you can get that. If you're going to use manure from a farm, or if it's your own source of manure from your own livestock or poultry, the system that you have for storing that and handling it is probably going to be fairly specific to your farm. So it's always good to also test the manure that you're going to be using. So really a good point.
Okay, great. This is more information about the talk from Cavagelli and Mirsky.
Yeah, I had a nice talk with Steve Mirsky just a month or two ago.
Yeah, he's a great guy. So as a member of Organic Valley, will you come out to help with agronomy, or can you recommend someone for us?
Well, if you're a member of our co-op, I'll be happy to help you. And actually, I'd like to help as many people as I can. So I want you to feel free to try to contact me, and I'll work with you as I have time. I have to focus my efforts on members of our cooperative, but I want to help people as much as I can. So by all means, let me know what I can do to help you. And if I can't, I'll try to put you in touch with somebody in your neighborhood who can work with you directly.
Yeah, and there's Matt Ruark. Dr. Matt Ruark is a soil scientist here at UW who does some extension as well, so he's someone you could contact, and I can put some information out about his work as well as a few other soil scientists.
Absolutely.
If a farmer had a good alfalfa plow down before corn, would applying manure add too much and bind up or suppress any other nutrients?
Well, it's possible. That's a really good question. you know, we've had this kind of a default mindset about manure for a long, long time. If we're going to grow corn, we just have to put manure on. Or conversely, if we've got manure, you know, it should go on the field where we're growing corn. Well, maybe and maybe not. And any time we get into excesses, those can cause as many problems as deficiencies. With nitrogen, you know, we've got this huge problem with this anoxic zone down in the Gulf of Mexico, and it's showing up in various areas, even in Green Bay, you know, in Lake Michigan. I mean, we can get this dead zone in there, and it has to do with nitrogen and phosphorus. And that stuff is getting in there because we're putting too much on the land. And everybody would love to get, you know, really wonderful corn yields. That's a good goal to have. But we also have to be content at some limit. And when we put nitrogen on to the point where the plants can't use it anymore and it's being leached down through into our groundwater, or when we put phosphorus on the ground to the point where, you know, every time we've got any kind of runoff, we're carrying a bunch of phosphorus off into some waterway, that's a big deal. You know, and we have to be aware of that. And as organic farmers, we're big on stewardship. You know, and we want to be good stewards of the earth, and we don't want to foul our nest or our neighbor's nest. So, yeah, too much nitrogen where we need it is not a good thing, and that's why I suggest accounting for the nitrogen that you can from the plow down first, and then just use manure for the extra that you need. A good question.
Right. From Bruce, I'm confused because the corn follows the alfalfa, which I thought would use up all the N. The second year after the alfalfa is in wheat. So is there a second year carryover from N from the alfalfa, or am I missing something?
Yes, there is. That's a very good question. And let me go back to that slide. How do we do that, Anders?
Just hit that drop down. You should be able to click and go to NEO.
Okay. I'm going to come back to the slide about legume credits. We just did not have a lot of time to look at this, but if you look at it closer, let's see, I'll try to use this star. Okay, over here on the left-hand side, on this column on the left-hand side, it says in the second cropping year following a fair or good stand on a fine or medium textured soil, you can take a 50-pound to the acre nitrogen credit. So what that means is that unless you got a soil that's a sand or a loamy sand, and unless you had what we would consider to be a poor stand of alfalfa, they're saying on average you should be able to get at least 50 pounds of alfalfa the second year following the plow down. And that's really most of what you need for a wheat crop even, you know, in most conditions. So I'd like to mention here too about how we define these things. Look at what a poor stand is. A poor stand they're defining is, I'll put the star over here. A poor stand they're defining is from 0 to 30% alfalfa, or less than 1.5 plants per square foot. That's really poor. And think about this, it does go down to 0. So what this means is that if you had a field that had alfalfa in it, and say you had a winter kill event or something like that, and all the alfalfa died, you could still go in there and plow that down and still get these nitrogen credits over here from that, because the alfalfa had been there and that stuff doesn't disappear instantly. Okay, so it's really substantial. So unless the stand is this poor here, you can credit 50 pounds of nitrogen equivalent the second year following the plow down. That's a good question.
Thanks for
okay, great. I had to fly through that because our short course guys were getting ready to leave there, but I'm glad you asked.
I think those are all the questions from online.
Okay.
If you have questions in class, you got a question?
Yeah, and this is actually partial comment related to this rotation discussion. But one thing I think to be wary of when you follow corn with wheat is if you're trying to grow for human consumption, there's been a correlation with those vomitoxin numbers going up when you plant wheat after a grass.
Okay.
So if you're trying to grow wheat for the human market, you want to plow down the legume straight into wheat.
Okay.
To try to keep that vomitoxin number lower.
Thank you. The comment was if you're going to be growing wheat for food as a food crop, there's been some correlation between manure applications and
no, it's
following the grass, following the corn. Some correlation with vomitoxin showing up more with wheat following a different grass family member. So that would be something to be aware of if you're growing the wheat for corn. But it's also important for livestock feed. I mean, we don't want to have mycotoxins in livestock feed either. So one way that you could get around that in some areas, depending on where you're farming and the growing season that you have, would be to break up the interval between the corn crop and the wheat with a cover crop. So what some farmers do is at the last cultivation, they'll go through and they'll broadcast a cover crop into the corn stand. Red clover, cereal rye, ryegrass, something like that. Ryegrass and cereal rye are still grass family members. But if you put in some clover in there, you could maybe break up that sequence, and maybe that would help with that. Or another thing that you could do is, if you're in an area where you can grow winter wheat, which, I can't up in my part of the state, but here you can grow winter wheat just fine. You could also interrupt that cycle. After the corn, you could come in with a spring cover crop and put something in quick, maybe some tillage radish and some red clover or some berseem clover or something like that. Get a quick cover crop in there just to interrupt that cycle. Then plow that down and put your wheat in following that. You still have time to establish a good crop. Thank you for pointing that out.
Yeah. My question was that beneficial relationship between the alfalfa and the bacteria in the soil. That's specifically to alfalfa with that, or does it have any clovers or legumes?
Almost all legumes have a relationship like this with bacteria, but they don't all use the same species of bacteria. So the family of bacteria that does this is called Rhizobium. And the species of Rhizobium that inoculates an alfalfa is also effective for sweet clover, but that's not the same one that red clover uses. And that's not the same one that bird's foot trefoil uses. So you have to be sure that you're using inoculated seed that has the correct strain of bacteria for that crop that you're going to be growing, unless you've had that crop growing there already and you inoculate, you use that seed that had been inoculated there, then you've probably got the soil colonized with that bacteria already. The bacteria that we use for soybean is just such a wimpy crop anyway, and so is the bacteria. So, we almost feel that you should just use inoculated soybean seed every time you plant soybeans, because it's probably going to be several years in between, and there may not be anything left by the time you get back to it again. Great question.